Hello, I'm Vivianna Onofrietti, and my project is the effects of coconut oil and algae on cariogenic pathogens. We're going to first start talking about dental caries, which is one of the most infectious diseases in humans. It is the remineralization and demineralization breakdown process and can consist breakdown of the enamel from the biofilm on the tooth surface. Next is Streptococcus mutans. And Streptococcus mutans are the main bacteria in the oral cavity that cause dental caries to form. Now that you've heard about Streptococcus mutans, I'm moving on to my second bacteria, Lactobacillus casei. Lactobacillus casei is involved with the secondary layer of the tooth, which is called dentin caries, and is also a probiotic used commercially to ferment dairy products. Moving on to extra virgin coconut oil, this is one of my testing agents I'm going to be using on the bacteria, and it is a rich source of beneficial medium chain fatty acid, which is availability to inhibit some species of bacteria, and it is one of the food containing the highest amount of lauric acid. My other testing agent is Spirulina platensis algae, which is a blue-green algae that contains 60% protein content, and it is an antimicrobial effect on certain oral bacteria causing gingivitis. Now moving on to my hypothesis. I tested if extra virgin coconut oil and Spirulina platensis algae have prolonged exposure to the cariogenic pathogens, Streptococcus mutans and Lactobacillus casei using minimum inhibitory concentrations, then the food derivatives lauric acid and gamma-linolenic fatty acids in both foods combine with inhibit the largest growth of both microorganisms because the cariogenic pathogens would be more sensitive against the synergistic effects of the fatty acids in the food than with the foods individually at greater concentrations. Moving on to my controls and trials, I did a positive control on the fluoride of the Streptococcus mutans and Lactobacillus casei and a negative control of the distilled water on the bacteria. The number of repeated trials were 6 per individual concentration and pure mixture, 6 negative controls, 6 for each of the 2 positive controls for a total of 120 trials. And for my materials, I used some things like some of my testing agents as you see listed there, petri dishes with sheep's blood, cotton swabs, an incubator with some micropipette, gloves, goggles, and a lab coat. For my procedure, I used the agar disc diffusion method, also known as the Kirby-Bauer method, where I took my blood agar petri dishes and streaked them with my 2 bacteria, Lactobacillus casei and Streptococcus mutans separately, and then I put the sterile paper discs on the inoculated agar with my sterile tweezers, and then I had made my concentrations, which was 6.5%, 13%, and 26% extra virgin coconut oil, and 4%, 8%, and 16% Spirulina platensis. Once I had my concentrations, I had loaded them with the micropipette on the proper sterile discs, both individually, then the mixtures of 50-50 concentration solution ratio, and placed them in the incubator for 24 hours at 37 degrees Celsius. When I took them out, I measured their zone of inhibitions with a millimeter measurements and a digital caliper and documented all of my data, which determined the resistance, intermediate or susceptible, for the Streptococcus mutans and Lactobacillus casei from acceptable agar disc diffusion zone diameters chart. These are some photos of my procedure where I streaked the bacteria onto the petri dishes and where I diffused the testing agents onto the discs, and then you can see in the bottom 2 pictures where I measured the zone of inhibitions with the digital caliper. In my data chart here, you can see I listed all of the food testing agents on the left with the mixtures and the controls. And on the right side, you can see the diameter of zone of inhibition, which I measured in millimeters, that show all of my 120 trials. Here's my bar graph where I measured the average zone of inhibition of Streptococcus mutans and Lactobacillus casei. I measured them individually and in mixtures, and as you can see, the mixtures were a lot higher than the ones individually for the zone of inhibitions, and is considered susceptible in the disk diffusion chart. So this is my ANOVA statistical analysis, where it shows a pairwise comparison concentrations between individual and mixtures. And on the right side, it shows my p-values to see if it is statistically significant. So with those pairwise comparisons, you can see here in my discussion, it was the largest significant difference between individual concentrations of extra virgin coconut oil with EVCO 6.50% LC versus EVCO 26.00% SM solution, which was p-value less than 0.0001. And it shows a significant difference. In the bottom, there's Spirulina platensis algae, which are the largest significant difference between the individual concentrations, which was of SA 4% SM versus SA 16% SM solution, which was p-value less than 0.0001. And then you can see the significant difference there. So now we're going to the discussion with the mixtures, starting off with the extra virgin coconut oil and Spirulina platensis algae. It shows the largest significant difference between the mixture concentrations of EVCO 13% and SA 8%, SA versus EVCO 13% and SA 8% LC solution, which show the p-value less than 0.0003. And you can see it shows a significant difference between the mixtures of concentrations. Going to the EVCO and SA concentration solution, the lowest p-value comparison, it shows the largest significant difference between individual concentrations from p-values less than 0.0001 to 0.0002 to 0.0003 at the bottom. And now for my conclusion. After all 120 trials, zones of inhibition surrounding all discs from all three concentration mixtures of both pathogens measured a range of 26 to 32.3 millimeters with the digital caliper, indicating from the agar disc diffusion susceptibility test chart that Streptococcus mutans and Lactobacillus casei are susceptible to the synergistic effects of EVCO and SP at lower concentrations than MIC when combined versus individually. There was a significant difference between all concentration mixtures with both pathogens according to p-values for statistical analysis of data in comparison to all individual concentrations, including the highest individual concentrations at double the MIC. It is conceivable that different fatty acids combined together could express a more potent antibacterial effect, and these combined effects could enhance an antimicrobial efficacy against both bacteria at sub-MIC to produce a greater antibacterial effect at a lower dosage to reduce the chance of the emergence of antimicrobial resistance bacteria strains. So for my future research, I considered experiment with additional oral bacteria involved in other oral diseases to examine the susceptibility to EVCO and Spirulina platensis algae. I'd also used different food oils to test their susceptibility to Streptococcus mutans and Lactobacillus casei. And I was also examining different concentrations of EVCO and Spirulina platensis algae to collect data on those effects of Streptococcus mutans and Lactobacillus casei oral bacteria involved in dental caries. Thank you very much for listening to my research.